Uh, Good evening, guys. Am I audible and is my screen visible? You can just write in the chat box if I'm audible or not, and if my screen is visible or not. Can someone please write in the chat box if I'm audible and my screen is visible? Thank you so much. Thank you, Sneha. Thank you, Shiny. Okay, I'll share my screen. Okay, so it's 6:05, so we can start. We'll admit people as and when they come along. So, good evening. My name is Aishi, and I am a PMRF research scholar from IIT Bombay, and I am a TA in this course, Applied Environmental Microbiology. And today we'll be discussing some of the sample problems, and you can ask me your questions, and if I am able to solve it, or else I'll look into it and let you know. And these problems will, this solving session will help you solve problems for this current year's assignment. So these are some practice problems that we'll solve today, and hopefully these will help you solve this week's practice problems. I think 8th February is the deadline. I was checking through the announcement sections. So let's move on to the first problem. Yes, so this is one of the practice problems that was there this year also, I think, in the week 0 assignment. So the question is, which of the following can be used in isolating bacteria with the target gene? So will you guys be writing in the chat section what do you think might be the right answer? We can proceed in that way. Which of the following can be used isolating bacteria with a target gene? Or one of you can unmute and also let me know, but let's do it one by one, otherwise there will be a chaos. So
Ma'am, selective screening.
yes, anyone else? It's already written. Okay. Do you know what the other options mean? Like selective screening, yes, it is the correct answer. But what is heterotropic plate count and biological oxygen demand, and what does dilution to extension mean? So when we'll be solving these MCQ problems, we'll not only try to answer the problem, we'll also try to learn what the other options mean. So I think that will help us solve the other questions easily, because if we know the definitions of the other points also, that will help us in other questions. So if we move on, so what does heterotropic plate count exactly mean? So that was the first option here. So heterotropic plate count is the standard plate count procedure that is basically used. You can say it's kind of a colony forming unit assay. It's a procedure that is used to estimate the number of live heterotrophic bacteria that require organic compounds of carbon and nitrogen for nourishment in the water. So this is how it generally happens. So you take the original sample, it can be any sample. Let's talk about drinking water. Let's talk about water analysis. So when we take water and this is the original sample, and we do a lot of serial dilutions. So you can see here 9 ml, 1 ml of the original sample we put into the second tube. Wait, I'll 1 second. Pointer. Yeah. So 1 ml of the original sample we put into the second tube, and the other 9 ml is saline. So basically we are doing serial dilutions at each step, and then we pour agar. Ludi agar is a solid medium on which bacteria grows. It's not a selective. Okay, 1 second. There are people joining. Still, sorry, there are people joining. I'll have to accept. Yeah. So selective. We are talking about Ludi agar. So we can pour agar onto Petri plates, and Ludi agar is not a selective media. So if we culture bacteria on it, any bacteria can grow, and basically Ludi agar can be used to check for contamination. Even when we are working in lab with our samples, we always streak on Ludi agar to check if our sample has any kind of contamination, because if there is, then we will be able to see on Ludi agar. And there are some other kinds of selective media that gives rise to only the kind of bacteria that we want to grow. Sorry for the interruptions, guys. I'll have to accept the other people as and when they come in. So these are the agar plates. So what happens in this procedure is that we take the original samples that we have taken for analysis, we do some kind of serial dilutions, and then we can pour in the agar plates. So obviously when we do the serial dilutions, whatever bacteria, whatever microorganism was going in the original sample, they'll be serially diluted. So 10 to the power minus 1, 10 to the power 0, like the original sample, the actual sample will have a lot of bacteria that will not be able to count. But as and when we do serial dilutions, 10 to the power minus 1, minus 2, minus 3, minus 4, by the time we reach minus 4, minus 5, we'll be able to count bacteria really well. So that is also visible in this sketch schematic diagram. So plate 1 has a lot of bacteria, but as we move on doing dilutions, we can see the number of colonies are getting less. So this is kind of a heterotrophic plate count that is used to see the number of bacteria. And this is another sketch that I thought I should add. So this heterotrophic plate count method was it's still very popular, but it was more popular way back when we didn't have a lot of technologies to rely on. Now there are much more advanced technologies that we do to count cells. We have the flow cytometry. Some of you might have done it, some of you might be knowing about it. Flow cytometry is a technique where every drop by drop analysis occurs. So if we can analyze the cell count, we can analyze the kind of color that each cell has. So it's basically used to do a lot of stuff. It's very what we call FACS as well. It's very advanced and it's most sensitive, reproducible, fast, and obviously automated. Everything happens. You just prepare the sample, you put it in the FACS tubes, and you use the instrument, and everything happens on its own. You can see on the computer the number of red colored, if you have added a particular dye, and if some particular cells are supposed to take that dye, then you can see how many cells are there in the total sample, how many cells have taken the dye, and etc. So this is a very automated procedure, but we are talking about heterotrophic plate count, and we still do it in the lab. But it's selective, variable, and very time consuming, and obviously it requires a lot of labor. So this was one of the options there. Obviously this is not the correct answer, but I still wanted everyone to know what is heterotrophic plate count. We might come across questions like this in the future. Yeah. So the other options was biological oxygen demand. So the other question was biological oxygen demand. So biological oxygen demand or biochemical oxygen demand basically represents the amount of oxygen that's consumed by bacteria and other organisms while they decompose organic matter under aerobic conditions. So aerobic means oxygen is present. So it's basically a quantifying factor. So the lesser the BOD level, the more pure the water is. So you can see if water sample has a very high BOD level, that means there's a lot of bacteria, microorganisms inside that is making use of the oxygen to consuming the oxygen basically. So that's how we see that drinking water has a BOD level of 1 to 2 ppm, while normal water might be 3 to 5 ppm, might be moderately clean. Drinking water should be more clean because we are consuming it, and polluted water has a very high range. It's like 6 to 9 ppm. So this is what biological oxygen demand means in a schematic diagram. And then we move to the other option, that was dilution to extinction. It is also quite similar to the heterotrophic plate count thing that we talked about. So here also we are doing serial dilutions. So there are dilution factors, and we are taking the original sample, we are diluting it, and then we are again taking some kind of volume and inoculating in another medium, and then we are incubating it. So then we are incubating it to see the kind of growth that happens. So the further dilutions should obviously show lesser growth. So the further dilution will obviously show lesser growth, and so as you can see from this, these are the black things show that there's probable growth in there, and these do not show that kind of growth. So we can start the dilution series again from C and repeat. So basically the entire process, the sample will have a lot of microorganisms. We cannot count that. So we are diluting that to, basically the name is very self-explanatory, we are diluting that to extinction. So we are keeping on diluting until we reach a single cell. So here we can see that C as the amongst the vials that have growth, C might be having the lowest amount of growth. From C, we'll start a dilution series again and repeat until a single colony is isolated, and then this result can be examined using epifluorescent microscopy. So this is the most labor intense, labor requiring process, I feel, and it takes a lot of, it should be taking a lot of time. Now we'll move on to selective screening. So selective screening can be a lot of types. So the main question, what was the question? Which of the following can be used isolating a bacteria with a target gene? So the answer is selective screening. Obviously the other three do not come close to the question at all. Those are just isolating bacteria from a sample or stuff. When we're talking about a particular target gene, we are moving to molecular biology. When we're talking about a particular target gene, we are moving to molecular biology. And the one selective screening that came to my mind was a blue-white screening. So if you know blue-white screening, great. But if you don't, I'll explain a little bit. So blue-white screening is a process by which we all know about plasmids, right? Plasmids is the extra chromosomal DNA that is present in bacteria. And plasmids are known to carry antibiotic resistant genes that we know. And generally when we work in molecular biology lab, when we do cloning and all, we try to, if we are trying to make a particular protein in huge amounts, the option we have is growing that in E. coli. E. coli is one of the most flexible bacteria around. So if you want to make various copies of a particular protein, we take the gene that is encoding that protein and we put that in the plasmid, and then we transform the bacteria with the plasmid, and we let the bacteria to grow. And then we can induce the expression of the protein using some kind of agents like IPTG, and we can induce the expression of the protein, and then we'll be able to, we are basically making the protein of our interest in bulk amounts. So what happens in this case, this is the plasmid, PUC18 is the name of the plasmid, APR is the antibiotic resistant gene. So the antibiotic resistant gene is very important for molecular biology techniques, because with this gene we'll be able to selectively screen for the bacteria of our interest. If suppose a bacteria has the kanamycin gene of resistance in the plasmid, and we want to isolate that particular bacteria, so when we are pouring agar, we'll add kanamycin, a working concentration maybe 50 microgram per ml kanamycin into the agar plate, and then we'll spread the bacteria or streak the bacteria, whatever you're doing. The cells which do not have the kanamycin resistance gene will die out, obviously, and we'll be selectively screening for the cells that have kanamycin resistance gene. So that is also a very simple form of selective screening, because if we are selectively screening for bacteria containing the kanamycin resistance gene. Also, if we are talking about a particular protein that has been genetically engineered into the system, then we can do this blue-white screening. So this might look complicated, but this is actually very easy if you're able to understand it. So this is the plasmid of interest that we are talking about. This is the plasmid, and this is APR is the antibiotic resistant gene, and this LAGC, and this is a multiple cloning site. So multiple cloning site, MCS, is basically a site in the plasmid that has restriction enzyme. We all know what are restriction enzymes. Restriction enzymes basically cut DNA at very particular specific sequences. They have very specific sites to cut DNA, and we have a variety of restriction enzymes that we can use. And when we are basically trying to clone a particular gene into a plasmid, we use restriction enzymes. So multiple cloning site has a lot of sites for restriction enzymes, of different restriction enzymes, and the LAGC is also present in this multiple cloning site. So if we are trying to incorporate a gene into the plasmid, we'll amplify the gene, and we'll cut the gene with a particular restriction enzyme. We'll also isolate the plasmid from the bacteria, and we'll also cut the plasmid with the same restriction enzymes. Okay, and enzymes in the sense that we have to worry about the specific sequence, not about sequence, specific sense in which the restriction enzyme works, because we need the gene to be inserted in a particular orientation. So if there's a gene, we'll cut one side of the gene with the restriction enzyme and the other part with another restriction enzyme, and we'll simultaneously use the same restriction enzymes on the plasmid as well, and then we'll incorporate the gene into the plasmid. So if the insert is our gene of interest, if the insert gets incorporated into the plasmid, then obviously the LAGC will be interrupted. The LAGC sequence will be interrupted, and the subunits, the lag C subunit will not be prepared. Only lag beta, which is already present, will be prepared. But if there is no insert, and if the plasmid has not been able to take in the gene of interest, then both the subunits of lag C will be prepared, both alpha and beta. In case of the other subunit, that is the beta subunit, is present in the genomic part of the DNA of the bacteria, genomic DNA of the bacteria. So both the bacteria will have the genomic DNA. So lag beta will still be prepared, but lag alpha, the lag alpha sequence is present in the MCS site. So if the gene of interest gets inserted into the MCS site, the MCS site will get interrupted, and lag C will also get interrupted, and hence will not see lag alpha being made. And when we'll plate these two bacteria finally on the agar will have X-gal. X-gal is kind of a substrate. That's the only thing you need to remember for now. X-gal is kind of a substrate that gets cleaved only when both lag alpha and lag beta is present. So in this case, when no insert is present, both alpha and the beta subunits will be prepared, and hence when they are plated on agar, the X-gal will be cleaved. The enzyme that is made will cleave the X-gal, and we'll see a blue colored colony. But in this case, when the insert is incorporated into the plasmid, the MCS site will be interrupted, the lag Z site will be interrupted, the alpha subunit will not be made, only lag beta will be made, and lag beta is not sufficient for the enzyme to function, and so X-gal will not get cleaved, and hence we'll see a white colony. So this is a very important selective screening process that is used to check for cells having plasmids that have the gene inserted. So this is basically a schematic sketch. You can always go through books, molecular biology books, to study more about this process. I personally find it very fascinating that plasmid vector and foreign DNA, we can check if there are white colonies, that definitely means the gene has been inserted. If there are blue colonies, that means either the insert is not there, or the insert has been or the gene of interest has been inserted in some other part of the plasmid that is not interrupting the lag C sequence. And this is a very important screening technique that we use to select a bacteria with the target gene. So the answer was selective screening, as someone answered, and this is a very beautiful technique to screen for. So we have the target gene and we can easily screen out a bacteria. So when we see this plate, we can take the white colony, because for sure we know that the white colony will have the gene. So we can take the white colony and we can grow the white colony in liquid media. So we are basically isolating bacteria with the particular gene, and then we are growing that bacteria. So this is a very important selective screening process. So we'll move on to the next question. So the next question is, mitochondria is known as the powerhouse of the cell. It is believed that mitochondria was an independent individual bacteria but was engulfed by another bacteria. Now mitochondria is an essential part of eukaryotic cell. The reason microbiologists believe this theory is because mitochondria. So there are 4 options. Will someone like to unmute and answer? Because I'm seeing the PPT slide, I'm not able to see the Google Meet screen. So if someone wants to unmute and answer, they can do it.
It has its unique genetic material.
Correct. So the other options, are they true, or are they true, or are they not relevant, not sufficient enough for the question? What do you think? Okay, the answer is correct. It has its unique genetic material, and that has actually confused some scientists when these things were getting discovered. This thing has confused scientists for, I think, years, because mitochondria is a separate organelle that we can see in eukaryotes, and still it has its own unique genetic material, which is, some people might say, not required, because we already have the nucleus. So why does mitochondria have a unique genetic material? So the answer is, it has its own genetic material. So I will look into the structure of mitochondria. So on the right hand side we have a mammalian mitochondria, and this image has been taken in a transmission electron microscope. So TEM, one second. I'm sorry guys, I really have to stop my PPT in the middle to accept people, so I'm sorry if this interrupts your thought process. So this, in the right hand side we can see this is the micrograph of a mammalian mitochondria, and this has been taken via TEM. Quantum imaging is a very sophisticated way to image, and it's very difficult to do electron microscopy on live cells because it basically hampers the cells. But this is a very good image, and we can see that in a mammalian lung cell, this is a mammalian cell, and we can see the cristae, the infoldings of the mitochondria in a membrane, and everything can be seen in the cross section. And in the left hand side we can see a cartoon schematic of the cell of the mitochondria, sorry. So mitochondria, so why do we think that mitochondria might have been independent organisms? Because they have a lot of features that other organelles lack, but mitochondria has. They have their own DNA, they do cell independent division, and their physical characteristics apparently is quite similar to alpha proteobacteria. And some mitochondrial genes, and the scientists have discovered that some mitochondrial genes have transferred to the nuclear genome over time, yet mitochondria still retain some genetic material for reasons not completely understood. So people are still doing research, so some mitochondrial genes have transferred to the nuclear genome as a eukaryotic cell got developed throughout the years. Some mitochondrial genes got transferred to the nuclear genome, so there was actually no point keeping a unique genetic material inside the mitochondria, but still mitochondria still retains some kind of genetic material for reasons that are not particularly understood till date. And the hypothesized transfer of genes from mitochondria to the host cell's nucleus likely explains so why mitochondria. So this is a popular opinion that since there was a transfer of some genes from the mitochondria to the host cell's nucleus, this probably explains why mitochondria can't survive outside the host cell anymore. So the scientists are of the opinion that since they have a unique genetic material of their own, they might be independent living organisms at some point of time during the process of evolution. But while you're moving forward in the process of evolution, some of the genetic material got transferred to the nucleus of the cell, of the host cell, and hence mitochondria can't survive without the cell support anymore. But still, this might be a clue that they used to survive at some point of time. So this actually goes back to the endosymbiotic theory. So the endosymbiotic theory basically it talks about infoldings in the plasma membrane of an ancestral prokaryote gave rise to endomembrane components including a nucleus and eukaryotic reticulum. So we know the prokaryote is a very underdeveloped version of the it's very different from the eukaryote, and it's not as well developed as a eukaryotic cell. So they are of the opinion that in a first endosymbiotic event, the ancestral eukaryote which was getting still developed, it consumed an aerobic bacteria that evolved into mitochondria. So they are of the opinion that an aerobic bacteria which used to live outside independently was consumed by a eukaryotic cell. We know bacteria are prokaryotes. So at some point of time, an aerobic bacteria was probably consumed by the mammalian, by the eukaryotic cell. At some point of time, they were probably consumed by the eukaryotic cell, and this evolved into mitochondria through the. This is very disturbing. And this developed into the mitochondria through the process of evolution. And in the second endosymbiotic event, they say that the early eukaryote consumed photosynthetic bacteria that evolved into chloroplasts. So in the plant cells, we have chloroplasts. So they are of the opinion that some photosynthetic bacteria might have been consumed by the eukaryotic cell, which developed into chloroplasts. So, and this gave rise to the modern photosynthetic eukaryote and the modern heterotrophic eukaryote. So this is the endosymbiotic theory that basically relates to the question that we were having, that which feature of mitochondria makes the scientists believe that it was probably an independent organism some time back. So yes, whoever said it is right. It has its unique genetic material. Out of all the options, that's the most correct and relevant for this question. It has its unique genetic material, and that's the reason the endosymbiotic theory was proposed. So we'll move on to the next question. So the next question is about Koch's postulates. So the Koch's postulates can be best explained as we have the 4 options. So I cannot see the chat box. I see some people might be writing in the chat box, but I cannot really see it because my screen is having the PPT. Can somebody unmute and tell which one do you think is the right answer? Anyone? Anyone? Are you writing in the chat box? I'll check once. Which one do you think is the right answer for the Koch's postulate question?
Ma'am, option A.
First,
Sorry, which one?
Option A.
logical step.
Logical steps for proving
Someone
a
is saying
causal.
logical step, someone is saying option B. Correct? Okay, I'll move on. I'll share why I think which one might be the right answer. So the right answer is the logical steps for proving a causal relationship between pathogens and a specific disease. That is the first option. So before that, we can talk a little bit about Koch's postulate and what it means. So the Koch's postulate were obviously formulated by Robert Koch and Frederick Loeffler. I'm not sure about the pronunciation. And it talks about 4 criteria that were designed to establish a causal relationship between a causative microbe and a disease. So just see the date. It's 1884 and 1890. That's way back. So when we talk about Koch's postulate today, it might not seem that significant in today's time because we have evolved and we are advancing in modern science very quickly. But just see the date and you'll realize how important and how very difficult these statements might have been at that point of time. At that point of time, coming up with a statement like this might have been very crucial and might be very important as well. So the Koch postulates are basically 4 criteria designed to establish a causal relationship between a causative microbe and a disease. So when I read about these postulates, I was like, this is very logical. What is there to provide? Like, why is it like a statement? But then I looked at the date, and then I was like, okay, at that point of time, this was a huge deal coming up with things like this. So Koch, the scientist, applied the postulates to establish the etiology of anthrax and tuberculosis, very popular diseases, but they have been obviously generalized to other diseases. So what do these postulates say? So I have tried to add a sketch here so that it's visually appealing and you can understand it better. So this rat is a healthy organism. I hope you can see my cursor. I'll try to, yeah. So this rat is a healthy organism and this is a diseased organism. So if you want to check what caused the disease or what happened, what different is there in this diseased organism that makes it sick, so we'll try to check the suspected, so we'll see the suspected causative agent must be absent from all the healthy organisms but present in all diseased organisms. So that is logical. If someone is healthy, the whatever is causing the disease should not be present in that person. Whatever is causing the disease should not be present in that person. So very logical. So for that, people might be taking fluid samples to test, blood samples to test. So these are red blood cell samples. So they have taken blood cell samples and they are testing it under the microscope to see if there is a suspected agent that can be seen under the microscope, whatever objective they are seeing it on. And then the causative agent must be isolated from the diseased organism and grown in pure culture. So now the second step will be to isolate. If we are seeing something different in the blood sample when we are comparing the healthy and the diseased organism, then we'll try to pinpoint that thing, that agent, and we'll try to grow it in a pure culture. So here, as usual, the healthy organism doesn't have anything different, so we have nothing to actually culture. But here we can see that along with the red blood cells, there are some green spots that might be a kind of causative agent causing the disease. So for that, we'll take the suspected agent that we can see and we'll grow it to a pure culture. So here they have basically streaked. I don't know if you people know about streaking. So when we streak a sample, we take some kind of bacterial culture and we streak it on a plate. We streak it on a plate in 4 quadrants. Our basic motive, our basic agenda is to get a pure colony of that sample. So when we are taking via a loop, there are a lot of cells, organisms on that loop, but when we keep on streaking through the 4 quadrants, the number of cells go lesser and lesser, and finally when we reach the 4th quadrant, we'll see a very pure culture. So you can see here in the diagram also that when they started streaking, there is a very heavy red color, but as and when they went on, we can see a pure colony. So we can, by this process, we can see that we can get a pure colony, because as we keep on streaking, we'll move to, it's quite similar to dilution concept. We'll move on, we'll keep diluting our sample and we'll get a pure colony. So the causative agent that we get from the diseased organism, we take that and we grow that in pure culture. We grow that basically, and then what we do to verify that this is the causative agent that was causing that disease, we will then infect a healthy organism with that exact agent. So So the cultured agent must cause the same disease when inoculated into a healthy susceptible organism. Now a diseased organism might be having two uh, harmful agents, we never know. So just by seeing one harmful agent, we cannot say that this is the exact thing that was causing the disease. So for this reason, we have to culture that agent, suspected agent, and we have to use that thing to inject to basically infect a healthy organism. And if that healthy organism gets sick, is diagnosed with the same disease, then we'll be able to say that this microbe is causing this particular disease. The same causative agent must then be isolated from the inoculated diseased organism. So the second time the infection that we do in the healthy organism, that organism should fall sick, should have the same disease, should be diagnosed with the same disease, and then we'll re-isolate that agent from that organism, and then we'll re-isolate that agent from that organism, and then we'll again grow it to see. So these are the Koch's postulates. So these basically try to relate a causative agent to a particular disease, and this was done in the 1884. So I hope you guys can appreciate that at that time coming up with something like this was not easy. So the first answer will be the correct one. The postulates can be best explained as logical steps to proving a causal relationship between pathogens and a specific disease. So the next question is, which of these cannot live if exposed to oxygen? Will anybody want to try? Does anybody want to try?
Facultative and aerobic bacteria.
Okay. Anything else? We'll try to learn what all these options mean. Then it will be easier for us to understand which one of the organism cannot live if exposed to oxygen. So the answer is, actually it should be obligate anaerobic bacteria. Basically when we are reading stuff like this, we have to see what obligate means and facultative means. But the answer will be opportunistic anaerobic bacteria, and I'll tell you the reason. I will learn a little bit about all the options that we can see here. So what are aerobic bacteria? Aerobic bacteria, it's very self-explanatory. Aerobic bacteria, there's air in it. So aerobic bacteria, the bacteria that survive and grow only in the presence of oxygen in the environment. So aerobes grow and live in an ambient air environment, 21% oxygen, the normal air that we have. So if we have to define aerobic bacteria, we can say that they are aerobic organisms that will not be able to survive in the absence of oxygen. Okay? So aerobic bacteria cannot survive in the absence of oxygen, and anaerobic bacteria do not require oxygen at all. So they are also referred to as anaerobes. So here I have tried to provide a difference, the number of difference between these two bacteria. So aerobic bacteria that thrive and grow with oxygen, anaerobic can thrive and grow in the absence of oxygen. Such bacteria cannot tolerate the presence of oxygen in their environment. And the cellular respiration in case of aerobic bacteria will be aerobically, obviously. For the anaerobic ones, it will be anaerobic. So these are some of the differences. Molecular oxygen will serve as the terminal electron acceptor. So these are very technical details. And for anaerobic, it will vary. Carbon, since those do not require oxygen, they can use carbon dioxide, sulfur, ferric, nitrate, fumarate. All of these can serve as electron acceptor. Aerobic bacteria will only survive in the presence of oxygen. Anaerobes cannot survive in the presence of molecular oxygen. In some cases, molecular oxygen is very toxic for such bacteria. Aerobes can be found in different areas like water, soil. Anaerobes, since they do not survive in the presence of oxygen, obviously they will not be exposed on the exposed surfaces like soil or some regions. They will be in oxygen deficient areas. So anaerobes generally, this will be anaerobes. Anaerobes generally localize themselves at the bottom of the liquid growth medium. So we have a liquid growth medium. If we are growing anaerobic bacteria, they will accumulate at the lower portion, because at the lower portion there is like no oxygen or very limited oxygen, so they'll accumulate there. Aerobic bacteria will be localized at the surface of the liquid growth medium, and these are some of the examples. So this also shows, so what are obligate aerobes? I'll come to the obligate and facultative term in a minute. So this shows where the bacteria will grow. So bacteria can be identified based upon their oxygen requirement, and depending on the oxygen requirement, bacterial populations will localize in different regions of the growth medium. So obligate aerobes, they'll require oxygen, so they'll accumulate at the top. Obligate anaerobes will accumulate at the bottom because they do not require oxygen, and sometimes oxygen can be really toxic for them. These are facultative anaerobes, these are aerotolerant anaerobes, and these are microaerophiles. So these are very different types. So I'll talk about the facultative anaerobes. Wait. So the obligate aerobes are organisms that cannot survive in the absence of oxygen. So these cannot survive at the bottom of the tube at all. So therefore obligate aerobes mandatorily need the presence of oxygen in the environment, and they use molecular oxygen as a terminal electron acceptor. They oxidize sugar and fats and generate ATP via the glycolysis, then electron transport chain, Krebs cycle. We know a little bit about Krebs cycle. We know a little bit about that. Little bit about that. The enzymes that are mostly involved in the respiratory chain of obligate aerobes are catalase, peroxidase, and these are very essential for aerobic biology. And some examples of obligate aerobes can be bacillus, mycobacterium species, pseudomonas. These are. Now we'll move on to facultative aerobes. Sorry guys, who's here? We'll move on to facultative aerobes. It's not shown here. Facultative aerobes are basically bacteria that do not entirely rely on the availability of oxygen in their environment. Instead, these bacteria can have other anaerobic methods as well to produce ATP or energy molecules. They can therefore survive even in the absence of oxygen. So, for example, by this I mean as opposed to obligate aerobes, facultative anaerobes do not only rely on oxygen. Oxygen is preferred, but even if there's no oxygen in the surrounding medium, they'll still be able to survive. They can take oxygen. They do not entirely rely on the availability of oxygen. They can also use anaerobic techniques to produce ATP. Or so the facultative term kind of means optional. That oxygen is fine, without oxygen is also fine. Microaerofields that we see over here, microaerofields need only a small amount of oxygen for the generation of energy. Okay, the presence of oxygen in higher amounts can be lethal, and that is quite self-explanatory over here. See, they are almost at the top of the medium, but they are still not at the surface. So they need a little bit of oxygen, but not a lot of oxygen. So being at the top might be harmful for them actually. Okay, what are aerotolerant anaerobes? Now, aerotolerants do not utilize oxygen for a metabolic activity or for the generation of energy, but they are also not adversely affected by oxygen. Like, even though they do not require oxygen for their metabolic activities, their oxygen is not also toxic for them. So it's fine. Either way, it's fine for them. And facultative anaerobes, so you can see facultative anaerobes can grow with or without, facultative anaerobes can grow with or without oxygen because they can metabolize energy aerobically or anaerobically. So facultative anaerobes, see here, so they are at the surface, they are almost at the surface of the medium. So they are utilizing oxygen, and that's not causing them any harm. So they can grow with or without oxygen, and they can do both. They can generate energy both both using oxygen or in other ways. So they gather mostly at the top because aerobic respiration generates more ATP than fermentation. So they also prefer accumulating at the top. So the question was, so this is the difference between facultative and obligate. So facultative refers to the ability to live under more than one specific environmental condition. It's less restrictive, it's more flexible. Facultative aerobe means that I can live with oxygen, I can also live without oxygen. Facultative anaerobe means I can live with oxygen, live without oxygen. I'll prefer oxygen because it's generating more ATP than fermentation, but without oxygen is also fine. But when we use the term obligate, it's basically restricted to a very particular environment. So the question was, which of these cannot live if exposed to oxygen? So accordingly, it should be anaerobic, opportunistic anaerobic. Wait a second. Sorry. So it should be opportunistic anaerobic bacteria. So we'll move on to the next question. Which of the statements are not true for a prokaryotic cell? Does anybody want to answer? Anyone? Which of the statement is not true for a prokaryotic cell? No one? I'm audible, right?
Yes, ma'am, you are audible.
Okay.
And
Does
I
anybody?
think prokaryotes are exclusively unicellular.
This is not true. That's what you're saying?
Yeah, that's what I'm
Okay.
saying.
Okay. Anybody else? So the more relevant answer is the mitochondria is the energy center for a prokaryotic cell. So if we see the prokaryote cell and the eukaryotic cell, we'll be able to see the difference clearly. So prokaryote is a very like not underdeveloped version of a cell. It's prokaryote basically means primitive, so it's very it's not as developed as a eukaryotic cell, and this is the eukaryotic cell. So mitochondria is a very prominent component of the eukaryotic cell, and prokaryotes lack mitochondria and instead they produce their ATP on their cell surface membrane. So these are the parts of the prokaryotic cell. This is also prokaryotic cell. So mitochondria is generally present in all the eukaryotic cells, and prokaryotic cells, since they rely on the immediate environment to obtain energy, they don't really have mitochondria, and this process, their energy acquiring process, is facilitated by various electron transport chains in the plasma membrane. And mostly they are single cell organisms like bacteria and archaea, and they're obviously less structured than eukaryotic cells. We can actually, it's very self-explanatory, we can see the cells and we can see how less structured, how less defined prokaryotic cells are in comparison to the eukaryotic cells. Also, they don't have a proper nucleus. They don't have a membrane bound nucleus like we have in eukaryotes. What they have is nucleoid. So their genetic material is actually coil shape and it's free floating within the cell. So we cannot call it a well developed nucleus. In case of eukaryotic cell, we have a nucleus, inside we have the nucleoplasm, inside we have the chromatin, there's a nuclear envelope, there's entire nucleolus, and the entire thing makes up the nucleus. But that is not present in prokaryotic cell. Prokaryotic cells have a very free floating genetic material, and the other thing is they also lack many membrane bound organelles found in the eukaryotic cells. They have the ribosome, of course, because ribosome is required for protein synthesis, but they do not have the ER, or they do not have mitochondria, they do not have chloroplast. So these are some of the membrane bound organelles that they lack. So prokaryotes have no mitochondria. So this was prokaryote is the energy center for a prokaryotic cell. So this is not true. Mitochondria is the energy center for a prokaryotic cell. This statement is not true. So I'll move on to the next question. Which of the following organisms are referred to as prokaryotes? Bacteria, fungi, protozoa, archaea. Anybody?
Bacteria?
Sorry?
Archaea.
Bacteria?
Okay.
Archaea.
Archaea. Okay. Okay. Anybody else? Or I'll move. So it's bacteria and archaea, because bacteria obviously we talked about right now, these are prokaryotes, and archaea, they also come in the prokaryote section. So we'll check what forms the prokaryotes and what forms the eukaryotes. So the single cell organisms of the domains bacteria and archaea are generally classified as prokaryotes. Pro means before and KRY means nucleus. It's basically before nucleus, which is very primitive. The animals, plants, fungi and protists are all eukaryotes. EU means true and are made of the eukaryotic cells. So protozoa, the other option was protozoa. Protozoa are also unicellular eukaryotes. As in all eukaryotes, the nucleus is enclosed in a membrane. So the bacteria and the archaea, these two form under the prokaryotes. The fungi, plants, animals and protists, this form under the eukaryotes. And this is a very concise way of explaining the difference. Prokaryotes are primitive, eukaryotes are very modern. Prokaryote structure is very simple, not developed enough. Eukaryotes are very complex. They have a lot of organelles. They have a lot of stuff happening in the cell at the same time. Prokaryotes DNA is in the nucleoid region, free floating DNA. There's no proper nucleus, proper membrane bound nucleus. It's not there. DNA is in the nucleus. Prokaryotes nucleoid is not membrane bound. DNA's nucleus is definitely membrane bound. There are no membrane bound organelles. There are membrane bound organelles in eukaryotes. Prokaryotes are smaller, eukaryotes are larger. So the answer will be bacteria and archaea, both come in the prokaryotes section. We'll move on to the next question. The culture independent sequencing based study of the genome from environmental samples is called metagenomics, metaproteomics, pathogenomics, functional metagenomics. Anybody?
Metagenomics?
Metagenomics.
Okay. Okay, so when we are solving questions like this, metaproteomics we can eliminate at the first step only, because it's talking about the genome. Proteomics talks about the entire protein repository. So we'll delete metaproteomics the first step. Other three, they are genomes. So what will be the correct answer? So it will be yes, metagenomics. So, what is metagenomics? So, metagenomics is basically the study of the structure and function of an entire nucleotide sequence, isolated and analyzed from all the organisms in a bulk sample. So, this is a very upcoming and hot topic right now. People are doing metagenomic study all around. So, they take samples in a bulk sample, maybe from a pond or anywhere, any water sample, and they do bulk sequencing, like the entire structure and function of the entire nucleotide sequences can be studied. Typically microbes, obviously, and the entire, so you can see in the sketch, they are taking the sample. There are a lot of different kinds of microbes in the sample, and they are observing the whole community. They are putting it in the sequencer. They are characterizing all the genomes. That's why it's called metagenome, and it's often used to study a specific community of microorganisms, such as those residing in the human skin, in the soil and water sample. I've not seen, I've heard people work on water samples. They take water samples from ponds and stuff, and then they do metagenomic studies on that. Apparently people do it on the human skin and the soil also, and this is a very up and coming field. So metagenomics research basically primarily focuses on the sequence based information, and only a small proportion is dedicated to functional genomics. So most sequence based information is called metagenomics. Only a small proportion is called functional genomics, and the functional metagenomics involves expressing DNA extracted from an environmental sample in a surrogate host to discover new metabolites and proteins. So our question was the culture independent sequencing based study. So that's how we can eliminate functional metagenomics, because functional metagenomics requires expressing the DNA, that we are taking an environmental sample, we are extracting DNA from that, and we are expressing that in a surrogate host to discover new metabolites and proteins. So this is what functional metagenomics means. So basically metagenomics, in this technique we are retrieving the entire microbial genome directly from the environmental sample, regardless of the nature of the sample and the abundance of microbial entities. You imagine if you take a cup, like a bottle of water from a pod, it's bound to have like many different kinds of microbes we cannot even think about. So it's like regardless of the nature of the sample and the abundance of the microbial entities that are in there, metagenomic analysis explores the entire genetic composition of the microbial communities by sequencing, and then they are doing subsequent analysis on that. So it also plays a role in understanding the biochemical part of the microbes in the atypical environments, as well as their interactions with the other environmental factors. And so I'll tell a little bit about a typical metagenomic analysis. So a typical metagenomic analysis involves the isolation of the metagenomic DNA from the samples, followed by RNA gene amplification, and then they do shotgun metagenome sequencing. They basically do sequencing. They do analysis of amplicon reads. They do analysis of metagenomic reads, and finally they determine the microbial community. They do a lot of in silico analysis. In silico is basically everything computational. They do a lot of taxonomic assignment, metabolic assays. They do a lot of prediction, interspecies analysis and everything, and then they with using these kind of fingerprints can be generated from the metagenomic analysis, and then they can do comparisons for the specific functional genes among the composite microbial communities. So it is a very and they also use a lot of reference databases. So there are a lot of genomic and these databases that they can use to align what they have got, and they can use the databases to see if these microbes have already been discovered, these microbes have already been sequenced before or not. So the answer would be metagenomics. Yes. The answer will be metagenomics. Yes. Next is soybean root nodules are an example of association between a higher ordered organism, plant, and a microbe. This type of association is called, anybody would like to answer? There are 4 options that we have here.
Mutualism.
Anybody else? Yeah, mutualism is the correct. Mutualism is the correct answer. Now we'll know why mutualism is the correct answer and what does this parasitic thing mean. So so this is an example of mutualism. So rhizobia is the kind of bacteria that live in the roots of legume plants, and they fix nitrogen. They fix nitrogen for the plants, and in return, and that's favor they're doing for the plant. So the plants are benefiting. So the bacteria is living in their root the root of the legumes, and still they're benefiting because the bacteria is able to fix nitrogen. And in return for the nitrogen, the bacteria is getting carbohydrates from the plants. So both the bacteria and the plants receive a benefit from this relationship. So if when the relationship is beneficial to both the parties, it's called mutualism. So soybean root nodules containing billions of rhizobium bacteria is an example of mutualism, because both the parties are like benefiting from this relationship. This is also another schematic. So the bacteria is residing over here in the legume roots, and the bacteria is fixing nitrogen for the plant, which the plant requires for its growth, and the plant is doing photosynthesis and all that things that it does, and then it is providing the bacteria with bacteria with food. So that's how both of these agents are helping each other. So this is called mutualism. But there is also another option which is called parasitism. So how do these two words differ from, how do these two concepts differ from each other? So when the kind of relationship is beneficial to both the parties, it's called mutualism. But when this kind of symbiotic relationship is only beneficial to one party and the other party is being harmed by it, that is called parasitism. If there's a bacteria that is just taking benefits from the host, maybe a plant or an animal, and it's not giving anything in return, and the host is being harmed by the presence of the bacteria, that will be parasitism. So at the expense of the host member, when one member gains benefits at the expense of the host member, that is called parasitism. So this the mutualism occurs between two non-parasitic species, and parasitism occurs between one is called the parasite, the other is called the parasite, the other is called the host. And in case of mutualism, both the organisms need each other. But in case of parasitism, obviously only the parasite is getting benefit from the relationship, so only the parasite requires the host doesn't require the parasite. And this is a very specific relationship, this is not very specific. So parasitism example, relationship between mosquito and human. So the mosquito comes and sits and basically it sucks blood. So the humans do not get any benefit from the mosquito at all. So it's a parasitic relationship. Lice on human, tapeworms in cow. All these are very parasitic relationships because only one party is benefiting from it. But in the other case, relationship between bees and flowers, it's a very mutualistic relationship because pollination happens, bees also get honey and everything. So these are very bacteria and humans, the digestive bacteria. So we have a lot of digestive bacteria in our gut, and those are very like helpful bacteria. We require those bacteria to digest our food, and they are also getting a lot of benefits by staying in the human body. So these are mutualistic relationships. There's another type of relationship that is called commensalism. This can come in another question, so I just wanted to explain this also. So mutualism is when both the parties benefit. Like example is sea animal and clownfish. So you can see both these smileys, both these are happy smileys, so both the parties are benefiting. So mutualism. Commensalism is like when one party benefits and the other party is not affected. It's not harmed, but it doesn't benefit at all. So barnacles stay in the body of whales, and I don't think the whales get any kind of benefit from staying from the barnacles. I'll have to read more about it. So this is commensalism. The species A benefits, species B is unaffected. And the third case, which is parasitism, like the tick is on the dog, one species is harmed and the other species benefits. So these are the three different types of symbiotic relationships that the most popular three types of symbiotic relationships. We'll move on to the next question. What is the unit to measure the length of DNA? Anybody?
Base pair.
Base pair.
Yes, RPM is obviously like revolutions per minute, so it's something that we use while doing centrifugations. So it's BP, like base pair. So there are a lot of units that we use to measure the length of DNA. Some kind can be linear measures, like we use nanometers, angstroms to talk about the length of DNA. We can use mass measures when people are conducting experiments in lab. We use mass measures like nanograms or Daltons. There are obviously cardinal measures like base pairs, nucleotides. So we know the DNA structure. We know that the nucleotides form base pairs. So that's what we always use. We can use compositional measures. We can use percentage of GC. So if you have done PCR reaction, you can see when you are designing primers for the PCR reaction, when you are designing it yourself, you have to calculate the percentage of GC in the primers to check if they will properly attach to your genome or not, but whatever you're trying to amplify. So that's also we use, very popular, percentage GC. The information measures bits, entropy, complexity, Then these are some of the cytogenetic distances also, we say centimorgans. It's a very popular unit also. Thermodynamic properties, these are not exactly units, these are properties, melting point, stacking energy. Then we have functional units. We know about genes, the introns, which are the sequences between the exons, not really required. Then the exons, we have the operator sequences, ORFs, transcripts, etc. So these are the various units that we use to generally denote our DNA with the nucleic acids. So the most popular one is the base pairs that you said right now. So one base pair corresponds to approximately 3.4 angstrom of length of the strand and to roughly 643 kilodaltons for DNA and RNA. And kilo base pairs is 1000 pp, mega base pairs is 1 billion pp. This is how. So for single stranded DNA RNA, units of for single stranded DNA RNA, obviously we cannot talk in base pair terminology, because base pair, the term comes because it's a double stranded sequence. So double stranded, one base from one strand and the other base from the other strand, they form base pairs. But in case of single stranded DNA RNA, we'll always use nucleotides. So, and the centimorgan I was talking, this unit is basically used to imply distance along a chromosome. And the number of base pairs is corresponds to it varies widely. In the human genome, the centimorgan is about 1 million base pairs. So it's just another kind of unit that we also use to denote chromosomes. So next question. Antonio van Leeuwenhoek invented a simple microscope and was the first to report V. animalculus. And the second point is, which was also the first time bacteria was seen under the microscope. And then we have 4 options. Which do you think will be the correct option? Anyone?
I think option B is correct, ma'am.
B.
First is true, but second is false.
Okay.
That's my take on that.
Anybody else?
B option.
B option. Even when I was thinking, I thought B might be the right answer, because I was of the opinion that Robert Hooke might be the first one to see microorganisms. But then I searched, and apparently Antonio van Leeuwenhoek, he invented the simple microscope, and he also, the correct answer is A. Even I thought it might be B, but Antonio van Leeuwenhoek, he invented the simple microscope, and he was also the first to report like microorganisms he saw under the microscope as well. So if we talk about Antonio van Leeuwenhoek a little bit, he's considered one of the first microbiologists. He's the father of microbiology, and he used to make optical lenses, and it is through his own microscope that he was able to see the microorganisms. So, and then he named them animalculus, which is the English translation of the Dutch word, which basically means small animals. And then he went on to observe various cells like muscle fibers under the microscope. So I got this from the internet. So this is apparently the microscope that Antonio van Leeuwenhoek used to work with, and just see the time, and just I cannot imagine how advanced people were at that point of time as well. I hope everyone can appreciate that, the kind of science that we have today, and we'll think that this is nothing. We have a lot of advanced microscopes today, but even at that point of time, with limited resources and with limited belief in science, I don't think people used to believe in science as much. With limited belief in science, there were scientists like them who actually moved forward, and they paved the path for science. So very deservingly, he's called the father of microbiology. So this is one image, painted image of Antonio van Leeuwenhoek working with his self-made microscope. So a little bit about Robert Hooke also, since I was also confused. So Robert Hooke was another scientist who observed microorganisms under the microscope, and he basically observed the honeycomb structure of the cork and named the compartments as cells. And he also contributed to the field of physics that we call Hooke's law, if you have heard, and astronomy. And the first microscope, this is a simple microscope that Antonio van Leeuwenhoek made, and another microscope was made by Hans and Zacharias Janssen. I think they were a father-son duo, and they are basically known to have invented the first microscope, and they were a spectacle maker, and they invented the microscope as well as a telescope. And if we move a little bit in this line, we can talk about Matthias Schneiden and Theodor Schwann, and basically they came up with the cell theory. They basically described the cell to be the basic structural and functional unit of organism, and they proposed that all living organisms are composed of cells, and these cells arise from cells that previously existed. So all these scientists have a very huge role in paving the way. So I think this will be the first one. Both 1 and 2 are true. So next question. Dash is when the nutrients from the environment are converted into new cellular material. Anybody?
Evaluation.
Evolution.
No, ma'am, it's anabolism.
Metabolism.
No, metabolism is the, ma'am, I mean, it's the both anabolism plus catabolism. And
Correct.
catabolism means breakdown, and
Breakdown.
anabolism
Correct.
is the
Correct.
integrating
Correct.
into new cellular
Huh.
material.
So basically, just remember that catabolism, whatever he said is correct. Catabolism is breakdown, anabolism is synthesis. So when it's written, nutrients of the environment are converted into new cellular material, that will be anabolism. Evolution is a completely different process. Evolution is how uh, like eukaryotes evolved from prokaryotes. Evolution is how human beings came to come by. So, people say that primates are very close to human beings, apes are very close to human beings. So evolution is a completely different process. Evolution is how whatever we see today, the more evolved versions of human beings or organisms came from what they used to be at some point of time. So evolution is completely different. Metabolism is both anabolism and catabolism, and anabolism is the synthesis process. So the answer will be anabolism. So if we talk a little bit about the difference between catabolism and anabolism, catabolism is a set of metabolic pathways that breaks down molecules. So you can see this is a molecule and it's breaking it down to smaller units and while releasing energy. So these catabolism, but just remember that catabolism means breakdown. That's it. That will be easier to remember, and that are either oxidized to release energy or used in other anabolic reactions. So these smaller units might be used in another anabolic reactions, another synthesis process, or they might be just releasing energy. Catabolism is like a set of metabolic pathways that construct molecules from smaller units, and these reactions require energy. So also obviously when you are taking small molecules and constructing a bigger molecule, this process will require energy. So it's an endergonic process. So this is another quite detailed example. So anabolism, as I said, anabolism is synthesis, catabolism is breakdown, anabolism is constructive phase of metabolism, catabolism is destructive phase of metabolism. And anabolism, since you're making stuff, you need energy, and catabolism, you're breaking stuff, you basically give out energy. So that means endergonic and exergonic. So these are some of the examples. Functional anabolism is functional resting or sleeping, functional body activities. So these are some of the examples that's there, but the main difference is anabolism is the synthesis process, catabolism is breakdown. So we'll move on to the next question. Match the microbes with their domain and habitat. Anyone? Anyone would like to try? It's completely fine if you don't get it right. Even I don't get it right, get things right at the first time. So just let me know. Just try to, the words are quite self-explanatory. So just think about what was the, when you say thermo, what is the first thing that comes to your mind? When I say thermo, the first thing that comes to my mind is temperature. So think according to that line.
Hyperthermophils archaea.
Which one are you saying? Hyperthermophile?
Is archaea. It's belong to the group archaea because they are resistant to the high temperature.
Okay, but there are two archaea. Which one do you mean?
First option.
I think option C is correct.
E is correct. Sorry, which
C,
one?
C. Option C.
1, 3, okay. 2, 3, 4, 4.
Third, hyperthermophile, the undersea volcanoes. Yes, ma'am. This one is correct.
Okay. So thermo means temperature. So think, if you're thinking according to that lines, it might be volcanoes. Then if you're going to alkalophile, obviously it talks about the pH.
Yeah.
If we go to barophile, baro is quite relevant to pressure anything. So that relates to pressure. So if you think about that along that lines, the accepted answer is yes, C. So 1 will be 3, bacteria sea ice. I'll talk about all these in detail. 2 will be 2, alkalophile soda lakes. Hypothermophile will be the undersea volcano because thermo is quite it's relevant to temperature. Barophile means it's basically pressure. We say 1 bar. Bar is a unit of pressure. So if we think according to that lines, barophile is pressure. So it's basically salt lakes or bacteria. So what are psychrophiles? So first of all, we have microorganisms at every part of the world at very extreme environments as well. So extreme environments means temperature, pH, humidity, salinity, sugar, everything. And it's quite amazing how microorganisms can survive at those extreme conditions. So psychrophiles literally means cold loving. So these are organisms adapted to growth at really low temperatures, having an optimum growth temperature of greater than 15 degrees Celsius and maximum growth temperature of greater than 20 degrees Celsius. So they have successfully colonized all the permanently cold environments from the deep sea to the mountain and polar regions. And some of the organisms, depending on the optimal growth temperature, are also known by the term psychotolerant or psychotroph, and they have basically successfully overcome two major challenges that arise during growth at low temperature. Because of low temperature, because of the decrease in temperature, it exponentially affects the rate of biochemical reactions, and second, the viscosity of the aqueous environment. So these organisms, they have successfully overcome these two things, and they can easily grow at cold temperatures. And obviously prokaryotic psychrophytes, they all of them do not have the same requirements. They vary in their requirements, and they have tolerance to oxygen and include aerobic, anaerobic. And how fascinating is that even psychrophytes can include aerobic, anaerobic and facultative species. So these are deep sea psychrophytes, for example, may require high pressures for growth, and if they require high pressure for growth, they can also be barophilic psychrophiles. So basically psychrophiles is the cold temperature one, so that matches with the sea ice, bacteria sea ice. Next we'll move to hyperthermophiles. Thermo means temperature, hyper is like extreme. So hyperthermophiles are organisms that can survive and grow at extremely high temperatures above 80 degrees Celsius. Okay. And hyperthermophiles are a type of thermophiles that can endure even higher. We call thermophiles, if we add hyper to it, that means these are even more tolerant to temperatures. So hyperthermophiles are, their optimum growth temperature is 80 degrees Celsius, and they're capable of withstanding very high pH and high pressure as well. And they mostly are found in hot springs and boiling steam vents where even moderate thermophiles cannot grow. And they mostly belong to the archaea group, with very few species belonging to the bacterial group. Archaea group can survive quite well in these extreme environments. And they have the one question that I had was, how do the proteins stay stable at high temperatures? Like at 80 degrees Celsius, how come the proteins are not degrading? So they apparently the hyperthermophilic archaea, they use structural based physical mechanisms to increase the thermostability of their proteins in the process of its thermophilic adaptation. And this stability of proteins and enzymes is improved as a result of an increased number of salt bridges. The cations that bridge charges between amino acid residues, so there are a lot of salt bridges, and apparently this feature helps in keeping the stability, keeping the protein stable even at that kind of temperature. And they have also a lot of ionic bonds between the positive and negative charges of various amino acids, and apparently this also stabilizes the proteins. Can you imagine proteins being not being degraded even at such high temperatures? And they are saying that the increased number of ionic bonds, it creates a very densely packed, highly hydrophobic interior of the proteins, and this resists the unfolding. At such high temperatures, the protein structure will unfold and get degraded, but this densely packed hydrophobic structure, it prevents the protein from getting unfolded, and hence so basically these organisms have come up with their own mechanisms to survive in these temperature. Next is barophiles. So like when I hear the word barophile, my mind goes to 1 bar, 2 bar, which is the unit of pressure. So these are defined as organisms that grow and thrive optimally at pressures greater than atmospheric pressure. So the term piezophile is also used to replace as a replacement to barophile, and they have been isolated from various deep sea environments. Obviously deep sea environments will be very high in pressure and have grown, and they'll be like those areas will be of low temperatures as well. So that's how barophile will be for bacteria and salt lakes. Like they are found at the deep sea regions, and they can survive in a very huge pressure thing. Alkalophiles are a group of extremophiles that can live and thrive in environments with extremely high pH value, like 9 to 13. That is very alkaline environment. So if we consider the pH meter from to 14, 9 to 13 is at the upper end of the pH meter, like pH range, and it's very alkaline atmosphere. And then there are of two types: obligate alkalophiles that grow in environments with pH higher than 9, and facultative alkalophiles. So here also, I talked about obligate and facultative. So obligate alkalophiles have very restricted range, so they can only survive with pH environments with pH higher than 9. Facultative alkalophiles, they can live at both at neutral pH and higher pH alkaline conditions. So this was the correct answer, was this one, C. Yes, C. Now on to the next question. Which of the following allows the transport of a chemical in and out through its membrane, acting like a protein anchor? Anybody? Okay, so the correct answer will be permeability membrane barrier. The protein anchor and structure and integrity do not really match quite well. So the permeability barrier is formed by lipids separating one aqueous environment from another, and this is one of the very prominent characteristics of biological membranes. And this barrier enables maintenance of different concentrations of solids on two sides of the membrane. So obviously the membrane that we have, some are selectively permeable membrane, some are semi-permeable membrane, and these help while these help in maintaining the structural integrity, while help in maintaining the integrity of the cells, these also help in maintaining a balance of the solutes ions at both sides of the concentrations on both sides of the membrane. So this answer will be the permeability membrane barrier. So next question is, in which of the following transportation mechanisms are periplasmic binding proteins involved and energy derived from ATP? Anyone?
I think diffusion transport.
Okay. Anybody else?
Binding proteins.
Diffusion transport.
Sorry, can you repeat?
Diffusion transport.
Okay. So the answer will be ABC transporter. I'll tell you why. I'll talk a little bit about all the options. So basically transport can be categorized into two categories, basically active transport and passive transport. Now whenever we're talking about active, it requires energy. Just remember that it requires energy. And passive transport is simple diffusion. Like simple diffusion, there's a membrane, there's a lot of ions and a lot of solutes on one side, there's a less number of solutes on the other sides. To balance both the sides, the solutes will move from an area of high concentration to an area of low concentration. This is simple diffusion technique. Then there is facilitated diffusion, which is either channel mediated or carrier mediated. So channel mediated diffusion will require a proper channel on the membrane, which will help in the passing of the solutes from the high to the low concentration. And facilitated diffusion will require a carrier, a protein carrier that will help in this kind of transport. If we move to the active transport requires energy. So whenever it requires energy, that might be against the gradient. So whatever we're talking about the passive transport, it was along the gradient. So things are moving from high concentration to low concentration. But whenever we require energy, just remember we are doing it against the gradient. So we need, in active transport there are also two types, primary and secondary. In case of primary, uniport. Uni is one. Uniport means only one particular thing is being transported. And see, it is being transported from its low concentration to its high concentration. This will not happen on its own. Things getting transported from low concentration to high concentration, basically against the concentration gradient, will not happen on its own. That's why it requires energy. So ATP is broken down to provide the required energy, and this solute or whatever it is, it's getting transported from its low concentration to high concentration. It's called uniport because uni is one, only one thing is getting transported. Then we have co-transport. In co-transport, is, you can understand, co means simultaneous basically. So in kind of co-transport, two different things are being transported, and both of these are getting transported from the low to the high concentration. Again, it's going against the concentration gradient, so it will require energy. So for these kind of transportations, there are various pumps. We have sodium potassium pump. We have various kind of pumps. If you have heard of sodium potassium pumps, sodium goes to one side and potassium goes to the other side. During any kind of neuronal signal at resting phase, one type of transport happens. When the neuron fires, another type of transport happens. So these are kind of co-transport. In the secondary transport mechanism, which are carrier mediated, we have antipode and sympode. In antipode, there are two things moving, but they are moving in the opposite direction. And in sympode, there are two things moving, but they are moving in the same direction. See this? This is going from high to low concentration. So this is going along the gradient, but this is going against the gradient, low to high. In case of antipode, this is going from high to low, this is going from low to high, but they are going in different directions. So this kind of transport is called antipode. So these are some of the definitions. Simple diffusion will mean movement of simple or lipophilic molecules which can easily go through the membrane. There will be no hindrance in the membrane. Osmosis basically is referred for water molecules, liquid molecules, and it depends on the solute concentrations. So if the solute concentration is more on one side, that means water will be less, the opposite kind of movement will happen. Facilitated diffusion will require any kind of large or charged molecules. You can see either it happens to a carrier or it happens to any kind of channels. So for these ions and sucrose, etc., for these large and charged molecules, ions are charged, these are large molecules. For these, any kind of carrier or channel is required. For active transport, obviously energy is required, and these are also primary, direct, or secondary indirect active transport. In case of primary, it involves the direct use of metabolic energy. Like direct use of metabolic energy, ATP is hydrolyzed, and that's how we get the energy. In case of secondary or indirect transport, it involves coupling the molecule with another moving along an electrochemical gradient. See, in this case, this high to low, this is occurring along the gradient. So these two molecules have been coupled. So this is moving along the gradient. That's how this molecule is able to move against the gradient. Similar for this. The antipode and sympode name is just given because in this case it goes to opposite directions. In this case it's going to the same direction. That's it. So this is that was active and passive transport. In case of, now we can talk about group translocation. That was another option here, group translocation. So what is group translocation? Group translocation is a mechanism that is widely used for transportation of sugars across bacterial membranes and perhaps those of some higher cells. Active transport is just something that goes across the individual of higher concentration assisted by enzymes and required energy. It's going against the against the gradient, concentration gradient, so it's called active transport. Group translocation is very specific to transportation of sugars across bacterial membranes, which are huge moieties. And the transporters in case of transport proteins, in case of active transporter, antiporters, sympoters, ATP binding cassette, which is ABC system. I'll talk about this. And group translocation. Group translocation is also a type of active transport. The transport systems very specific to group translocations are PTS system or the phosphotransferase system. And as I talked about active transport, ATP is getting hydrolyzed in case of active transport, so uses energy from ATP or the electrochemical gradient. Here energy is taken from ATP, here there's an electrochemical gradient that is being followed in one of the solutes, so that is a source of energy. In this case, group translocation, it is breaking down high energy compound like PEP. PEP is phosphoenol pyruvate. It is a very high energy compound. It is breaking down this compound to get the energy. For active transport, it moves glucose, amino acids, ions. Ions are charged, glucose, amino acids, they are also moving. Group translocation, as I said, these are very specific to sugars. They transport many sugar like glucose, mannose, fructose, all of these are sugars. And then active transport, it releases exactly the same molecules that have been taken up from the medium. For group translocation, molecules which are taken up from the medium undergo modification. They might undergo modification. These are the differences between active transport and group translocation. The other option was ATP binding cassette, which is the correct option. It is also called the ABC system. So example of an ATP dependent active transport can be found in gram-negative bacteria. So this involves substrate specific binding proteins located in the bacterial periplasm, the gel-like substance within the bacterial cell wall and cytoplasmic membrane. So this is how the ABC system works. There is a nucleotide binding domain at this end and there is a solute binding domain at this end. And this is how the ATP binding cassette works out, and it is a very one of the examples is how in the gram-negative bacteria this works, and you can see the periplasm. The question was periplasmic binding proteins involved. Okay, so how does this work? So the periplasmic in case of the ABC system, the periplasmic binding protein picks up the substance to be transported and carry the membrane spanning transport protein. So this is the membrane. This is the phospholipid bilayer. This is the membrane transporter, basically transporter protein. This is the periplasm region, and this is the substance that needs to be transported to this side. So the periplasmic binding protein, this protein attaches to the solute here, and then it takes it to this transporter protein. Okay, this is the first step. In the next step, the molecule that is to be transported across the membrane enters the transporter protein, and a molecule of ATP enters the ATP binding site of the ATP hydrolyzing protein. So here we can see this is the ATP, this is the transporter protein, this blue one, and the purple side is the ATP hydrolyzing protein. Okay. So once this solute, which is to be transported, once the solute is being pushed inside the transporter protein to be shifted to this side, ATP comes and attaches to the ATP hydrolyzing protein. This happens the second step. Simultaneous happening these are. Then third step, energy provided, the ATP binds to the ATP hydrolyzing protein and it gets hydrolyzed to ADP and inorganic phosphate. ADP and phosphate. So ATP was here, it got hydrolyzed to ADP and phosphate. And simultaneously the energy provided by this hydrolysis process, the energy provided moves this molecule from the transporter to this side across the membrane. So you can see how these two processes are very simultaneous. ATP gets hydrolyzed, energy is being provided, substance, the solute which was carried to the transporter protein by the periplasmic binding protein, which was in the previous periplasmic binding protein, it moves to the other side. This is the third step. And the fourth step, the carrier protein releases the molecule being transported and the transporter system is ready to be used again. So the protein was in this area, so it releases the protein to this side and then the system is again ready to be used. This is a kind of a cycle system. This is called the ATP binding cassette. That is why it is called the ATP binding cassette, because there is ATP is binding. So the periplasmic binding protein is coming, it's taking the solute inside, ATP is getting hydrolyzed, energy is produced, ATP is getting hydrolyzed to ADP and PI, solute is being shifted to the other side of the membrane, and the transporter protein is ready to be used again. So that is why the question was, in which of the following transportation mechanisms are periplasmic binding proteins involved and energy derived from ATP? So that will be ABC transporter, ATP binding cassette. This is the ATP binding cassette transporter. So that's why the answer will be ABC transporter. Okay. Then move on to the next question. Which of the following statements are true? Phase contrast microscopy scans the surface of the sample with the probe and maps the topography. You guys read and let me know if you want to unmute and let me know the answer. Which one might be the right answer? These are the 4 options. Anyone? I'm audible, right? Sorry. Yeah.
Ma'am, last option.
Last option is the right one. Right one. That's what you're saying, right?
Yeah.
Anyone else? Yeah, you're correct. The last one is the right option. And we'll discuss a little bit about all these microscopy techniques. And I'll tell you, all these microscopy techniques are very relevant and they are very prominent in today's research. Especially confocal microscopy is very relevant for live biological samples. Confocal microscopy is like very important. It is indispensable. We cannot do without confocal microscopy. Electron microscopy, atomic force microscopy, and phase contrast are also very important techniques. So I'll talk about these techniques in a little bit. So what is a phase contrast microscopy? The name is quite self-explanatory. Here it is written, phase contrast microscopy scans the surface of the sample with the probe and maps the topography or height of the force. This is a wrong statement because this job is done by the atomic force microscopy, which I'll discuss. So phase contrast microscopy doesn't do this thing. So it's a false statement. So that's what does phase contrast microscopy do? So it's an optical microscopy technique that converts phase shifts in light passing through a transparent specimen to brightness changes in the image. So obviously if we have a specimen and there will be different parts of the specimen. So one part of the specimen will be more dense than the other part. So there will be a difference in how light passes through the 2 parts of the specimen. And this thing is utilized by the phase contrast microscopy, and then they utilize it and give a visual readout. Like if a specimen has 2 parts, one part is more dense than the other part. The other part, the less dense part, will easily allow light to pass through, and this difference in the 2 specimen parts is what is being utilized, and they show it to us by changing the brightness basically. So phase shifts themselves are invisible but become visible when shown as brightness variations. So we cannot really see that which specimen part is more dense and which is less dense, but when this thing is utilized to show a change of a difference in the brightness, that's how we'll be able to see. So this is phase contrast microscope. I got it from the internet, and this you can see this image, and this is how you'll be able to realize these are the same cells, the exact same specimen that has been imaged with traditional bright field microscope, this one, and then with phase contrast microscope. So you see how clear the clarity of the phase contrast microscope. So this is the phase contrast microscope imaging. So it's a very, very important microscopy in today's date. The next option was atomic force microscope or scanning force microscopy. So this atomic force microscopy has a cantilever. Basically, they have a cantilever, and this cantilever scans through the entire sample. If you have a cell sample or a bilayer sample, whatever sample you have, it scans through the entire sample, and the probe. So basically the AFM generates images by scanning this small cantilever over the surface of the sample, and this sharp tip on the end of the cantilever, this is the sample surface, and this is the tip of the cantilever. The sharp tip of the cantilever contacts the surface. So obviously the surface is not flat. It's obviously not flat. The sample will have bends and will have curves. So the sharp tip on the end of the cantilever contacts the surface, bending the cantilever and changing the amount of laser light reflected into the photodiode. The height of the cantilever is then adjusted to restore the response signal, resulting in the measured cantilever height tracing the surface. So as and when the cantilever, when it scans through the surface of your sample, when there is a curve, when there is a bend, cantilever will dip in accordingly or move up accordingly, and this will come out as a readout reflected into the photodiode. And this is how the atomic force microscopy works, and this has demonstrated resolution on the order of fractions of a nanometer, more than 1,000 times better than the optical diffraction limit. So by atomic force microscopy you can actually do a lot of stuff, not only in biology, in metallurgical material sciences also. AFM is a very important technique, and you can actually figure out the elasticity of sample, the basically how the sample structure, the height of the sample, which part is above, which part is below. You can figure out a lot of things from AFM. So the technique might sound simple, but you can actually analyze a lot of stuff about the sample from AFM, or atomic force microscopy. The next is the confocal microscopy. It is, according to me, one of the most important microscopy techniques required for biological samples. If you are a biology student, if you are doing molecular biology, microbiology, you have to do confocal microscope. And why I'm saying this, you can see the difference in these images. The same image is wide field fluorescence microscope, and this is a confocal microscope. And why does the confocal microscope show such clear images? There's a reason behind that. There's a logic behind that. So confocal microscopy is also called confocal laser scanning microscopy, and it's an optical imaging technique for increasing the optical resolution, contrast, clarity of a picture by means of using a spatial pinhole. So if you see the microscope over here, there's a pinhole over here, there's a pinhole over here, there's a pinhole over here. These are the, when somebody asks you what is a confocal microscope, how does it work, this is the most important part that you have to say, because confocal microscopes have pinholes. The pinholes basically block the out of focus light. In the wide field microscope, there's a lot of out of focus light that is falling on the sample, and the images are coming out like this, blurred and not clear. But the pinholes here, they bar out, they block the out of focus lights, and capturing multiple two-dimensional images at different depths in a sample enables the reconstruction of the three-dimensional structures, also known as optical sectioning within an object, and this technique is extensively used everywhere. So what is optical sectioning? So there's a sample, we all know about the x-axis and the y-axis. When we put a sample under the microscope on the stage, we can shift the field, we can see that we can change the x-axis, we can change the y-axis. But what about the z-axis? What about the depth of your sample? What about if you have a cell sample, where exactly on the in the depth of the sample is your cells located? That is what is called optical sectioning. That is something that confocal microscope do very well. So you can see there is a confocal pinhole. So when you are focusing on a particular area, only that area will be under focus, and all the light from the out of focus area will be blocked, and that is how you get such clear images. And then you can change the Z. Just like how you can change the X and Y on the microscope by changing the stage area, you can also change the Z axis. You can also Z axis. Sorry, you can also change the Z axis, and you can basically change the Z axis. You can focus on different Z axis, basically various planes of your sample, and you can create an image like that. There's one plane of your sample, the second plane of your sample, and you can basically do optical sectioning. At each moment you can do different Z stacks, and then you can get a 3D image. You can actually see on the microscope a 3D image of your entire sample. So confocal microscope is very important. If you like, I would suggest you guys read it or see YouTube videos. There will be excellent pictures of confocal, how confocal works and why it's so important. Basically because of the pinhole, it has become a lot more clear. The other one is the electron microscope. So the right answer was electron microscope. Electron microscope is a technique that helps us in getting high resolution images of biological and non-biological specimens. But still, biological samples getting electron microscope is not really compatible with biological samples, because it's the samples, these are live. If they are live cells, those require to be treated with very gently. Electron microscopes are very strong because we have the electron. There's an electron beam. It's not light. Even for the other microscopes, when we put a lot of, for confocal also, when there's a lot of laser light, the sample gets bleached, and the fluorescence, there's no more fluorescence, cell might get damaged. But still, confocal microscope works out. But electron microscope, it is very difficult, I feel, to work with electron microscopes for cells, because it damages the specimen. But then since it gives such a high resolution because of the electron beam, it is also used to investigate detailed structure of tissues, cells, organelles. So live cells cannot really be imaged. When you are fixing cells, dead cells, can be imaged really easily in the electron microscope. Live cells, it will be I don't think live cells can be imaged in electron microscope. I'll have to see if anybody has tried it. So this is how electron microscopes work out. There are basically two types of electron microscopy technique: the scanning electron microscope and the transmission electron microscope. So from the name you can see the scanning electron microscope will basically scan the surface. You'll get a very good surface image. The transmission electron microscope will go inside and will give a better basically go inside the sample and will give a better image. So according to that, this option, the electron microscope uses a beam of electrons and their wave-like attributes to see with very high resolution. This was the correct answer. The other answers are not correct. So yes, move on to the next question. The primary differences between archaeal and bacterial cell membranes are: bacteria have ester linkages with glycerol and archaea have ether bonds. One second. Someone is joining now. So the primary differences between archaeal and bacterial cell membranes are: bacteria have ester linkages and with glycerol, and archaea have ether bonds. And the other point is bacterial membranes have rings and sugars, unlike archaeal membranes. So does anybody want to answer this question? Which one is the correct option?
A is true.
A is true. Okay. Anybody else? Anybody else has the same answer or different answer?
Ma'am, both are true.
Both are true. Okay.
B option.
Okay, you are saying both are true. Someone is saying A is true. Okay, we'll move on to the answer. So A is true, and for this we really need to have a little bit of a knowledge about the cell walls. The cell wall, we'll talk a little bit about the cell wall and the difference between archaea and bacteria. So this is an archaea. This is even less developed than a bacterial cell, and this is a bacteria. So these are some of the differences between an archaebacteria and eubacteria. So eubacteria are more complex than archaebacteria. Archaebacteria, as I said in one of the questions, they are found in very extreme environments. Even though they are very simple in the organization, it's fascinating how they have managed to survive in such extreme environments. How they can survive in hot springs and cold temperatures. It's really fascinating, even though they have the simplest of the structures. Archaebacteria can be spear-shaped, rod-shaped, plate, spirals, even square-shaped. And eubacteria are the normal shapes that we have talked about: cocci, bacilli, vibrio, rods. Now coming to the cell wall of archaebacteria, the cell wall of archaebacteria doesn't have peptidoglycan. It has something called pseudopeptidoglycan. The basic difference is this only, because the bacterial cell wall is very highly enriched in peptidoglycans and uramic acid, but the archaeal cell walls do not contain this compound, though some species may contain a very similar compound. This compound is called pseudopeptidoglycan. Now, what is the pseudopeptidoglycan? This peptidoglycan is a substance that is chemically very different from peptidoglycan, but structurally and functionally very similar. Even though chemically it's different, structurally and functionally it's similar. So this is one of the differences in the cell wall. The other differences: introns are present, introns are absent. These are some of the differences. If we move on to the cell wall difference, in the second last difference, we can see that membrane lipids are ether linked branched aliphatic chains containing D-glycerol phosphate, and in case of eubacteria, membrane lipids are ester linked straight chains of fatty acids containing L-glycerol phosphates. So it is likewise, archaebacteria also do not have cellulose or chitin. The cell wall of archaeans is chemically very distinct, and the most striking difference between archaea and other living things, it lies in the cell membrane. As I said, there are 4 fundamental differences, and those are the chirality of glycerol, the ether linkage. The option had ether linkage. See, bacteria have ester linkages and archaea have ether bonds. So this ether bonds, that's why A option is true, because there are ether linkages in archaea, and they have isoprenoid chains and they have branching of side chains. And in archaea, there are like ether bonds between the glycerol head of the lipid and the tail. And in bacteria, the tail of the lipid is composed of fatty acids, but in archaea, these are replaced by repeating units of an organic chemical called isoprene. That's why there was somewhere that said it has isoprenoid chains. So the main difference between these two things, archaea and bacteria, are the cell wall. One has peptidoglycan, one has pseudopeptidoglycan, and one has ether linkages. The other prefers ester linkage. That's why the option A was correct. Okay. This is a very theoretical question. There's no concept in it. You just have to study and remember. So that's it. You can just search Google also the differences between the archaeal and bacterial cell membranes. The next question is translation is synthesis of proteins by ribosome, and topoisomerase helps in unwinding the tightly wound double helix structure of DNA. These are two statements. Which statement do you think is true? Which one of the four options is correct? Anybody?
A and B both are independently true statements.
Okay. Anybody else? Yes, you're correct. Both A and B are true statements and they are independently true. They have no relationship to each other whatsoever. So that option is C. A and B are both independent true statements. So little bit about translation. I'll not go into the detail. So you can check in any molecular biology books what translation means. So basically it's a process in which ribosomes in the cytoplasm or the ER, endoplasmic reticulum, they synthesize proteins after the process of transcription of DNA to RNA. So if you see the concept of central dogma, the concept of central dogma states that there is DNA which is the primary genetic material. DNA is duplicated. The DNA replication is a semi-conservative mode of replication. The DNA is replicated and then from DNA RNA is formed, which is a single-stranded mRNA, messenger RNA. That process is called transcription. And from mRNA the proteins are formed and that process is called translation. So translation is basically mRNA or messenger RNA to protein. Okay, so whatever genetic information was there in your DNA, it's getting transferred to mRNA and it's getting transferred into your proteins. The entire process is called gene expression. So in translation, the messenger mRNA, this is the messenger mRNA, is decoded in the ribosome. Ribosome is a very important organelle in your cells. Even prokaryotes have ribosomes. One second. Yeah, so ribosomes are very important. I feel one of the most important because it's helping synthesize protein. What else is required? So the ribosome comes to the messenger RNA, and everything this happens in the cytoplasm, not inside the nucleus. The nucleus mRNA comes out of the nucleus. DNA is inside the nucleus. DNA is converted to mRNA. mRNA comes out of the nucleus, and then translation. The mRNA and the ribosome, both of these work together to produce a specific amino acid chain or a polypeptide or then a protein. The polypeptide is basically a chain of proteins, a chain of these things, amino acids, and with a peptide bond. And when this polypeptide later folds, we know there are a lot of structures of proteins: primary, secondary, tertiary, quaternary. So when this polypeptide folds, that's how a protein is formed. Okay, polypeptide and protein is essentially almost the same thing. I shouldn't say the same thing because a polypeptide is only a chain-like structure, but a protein needs to have a tertiary and quaternary structure to be called a protein. And then this ribosome facilitates decoding by inducing the complementary tRNA anticodon. So this is a very complex process. This image is also complex, and if you check in any molecular biology book, this process, the translation, transcription, all these processes are a very complex process because it requires a lot of proteins, a lot of things, and everything needs to happen in a particular sequence. So for now, just remember the translation. For this question, what we need to know is the translation is mRNA to protein. And tRNAs, which are transfer RNAs, transfer mRNA has the information of a genetic material. tRNA or the transfer RNA, they basically bring specific amino acids to be added to the polypeptide, and the mRNA passes through and is read by the ribosome. This is the mRNA, it is read by the ribosome. These are the specific tRNA that are bringing, these are amino acids attaching to the tRNA, and then the tRNA are bringing the specific amino acids. So the mRNA is here, it is reading a code, and the amino acid corresponding to the code is being brought by the tRNA. So it's a very complex process. You can check it out. For this question, just remember the translation is mRNA to protein. So translation is synthesis of proteins by ribosome, which is the correct statement, as she said. A is true. And what is topoisomerase? So DNA topoisomerases are very ubiquitous enzymes. They are found almost everywhere, and they are found in all cell types from viruses to man. So ubiquitous means they are very prominent in various structures. So they are even found in viruses, and they are found in humans. So these enzymes act to regulate DNA supercoiling by catalyzing the winding and the unwinding of DNA strands. Now when DNA is being replicated, there needs to be a replicated, any other process is going on, the DNA needs to be unwound and then it needs to be wound back. So this is the unwinding is caused by topoisomerase. So here you can see topoisomerase is a protein that binds to the double helix ahead of the replication fork. The replication is happening. This is how the replication is happening, and relieves the strain placed on the double helix as it unravels. So we know that DNA has two strands, and the strands need to unravel, and then from one side the complementary strand is being synthesized of one strand. So here the strand has unraveled and this new strand is getting prepared, and basically the job of the topoisomerase protein is to unwind this double helix. This is how it's moving forward. The DNA helix breaks the hydrogen bonds between the two strands, and this allows the DNA to unwind into two strands. And then there are also single strand binding proteins which prevent the separated strands from joining together again by binding. The single stranded binding proteins basically stabilize the process. They bind to the single strand portion of the replication fork, and they prevent. Since they are complementary bases, they'll also, even if they're separated, they'll come back together really fast. But we need to separate them for the replication process to occur. So these single stranded binding proteins keep these separated, stabilizes the process. They stabilize the process and allows replication to go through. So for this question, topoisomerase, it helps in, it acts to regulate DNA supercoiling by catalyzing the unwinding and the winding of DNA strands, which is true. So both these statements are true, but they don't have any relation to each other. So these are very true, independently true statements. So I'll move on to the next question. Dash is a mixture of salts, sugars, amino acids, nucleotides, and many other biomolecules. What do you think the answer is? Anyone? Anyone would like to answer?
Cytoplasm.
Correct. So cytoplasm is a mixture of salts, sugars, amino acids, and nucleotides. So what is a plasmid? Cannot be a mixture of salt, sugars. Plasmid is basically DNA. It's just not genomic DNA, but it is DNA. So what is plasmid? Plasmid is a small circular double-stranded DNA molecule that is distinct from a cell's chromosomal DNA. So this is a bacterial cell. This is the chromosomal DNA. These are plasmids. These are also nucleic acids, DNA, but these are not genomic DNA, and they naturally exist in bacterial cells. They also occur in some eukaryotes, and the genes, as I said in another question, these carry antibiotic resistant genes that provide bacteria with genetic advantages such as antibiotic resistance. This is a plasmid. So if you are interested, or if molecular biology is something that fascinates you, you should definitely read more about it, because these things are not only theoretical. We work with these things in the lab. So if you want to produce a protein, we actually insert the DNA encoding that protein. We insert that DNA into the plasmid, and then we transform the bacteria. We take out the plasmid from the bacteria first, then we insert our DNA, what we want to incorporate. For example, insulin. We want to incorporate the gene encoding insulin, and we add that to this plasmid. Then we put the plasmid back into the bacteria, and then we allow the bacteria to grow and grow. And E. coli divides in 20 minutes. So you can imagine in one day, like within 16 hours, there will be a lot of bacteria, and then we can isolate protein from that. A lot of bacteria producing insulin, and then we can actually take out a huge quantity of the protein insulin from there. So this, if you are interested in genetic engineering and molecular biology, this technique is cloning and everything. It really fascinates me. So I would ask anyone who's interested to read more about it, what plasmid does and everything. So the other thing was ribosome. Obviously ribosome is a mixture of protein and RNA. It's an intracellular structure made of both protein and RNA, and it's the site of protein synthesis. So this is a small subunit and large subunit. This is the messenger mRNA I was talking about, and it reads the mRNA sequence and translates the genetic code into a specific string of amino acids, which grow into long chains, then form proteins. So this also has only RNA and protein. The question had salts, sugars, and everything. So obviously this is also not the correct answer. The next is DNA. DNA is the most basic structure that we have learned about. DNA is a deoxyribonucleic acid. It happens in almost every cell. It is there in every cell, and we have the four bases: adenine, guanine, cytosine, and thymine. And adenine base pairs with thymine, and guanine base pairs with cytosine to form units called the base pairs. So these are nucleotides that are arranged in two long strands, and it forms a spiral ladder kind of shape. And the ladder's rungs are basically the base pairs. And an important property of DNA is that it can replicate and make copies of itself. So even DNA is not the correct answer. What is cytoplasm? But cytoplasm, that is a very common feature of all the cells, plant cell, animal cell. Cytoplasm is everything outside the nucleus. you know where other organelles are there. These organelles are present in the cytoplasm. If this is the nucleus, these are the other organelles. So cytoplasm will have a mixture of a lot of things. So obviously a mixture of salts, sugars, amino acids and nucleotides. Amino acids, everything can be found in cytoplasm. The other options naturally cancel out. Cytoplasm is the correct option. So the other next question is, the sequence of 3 nucleotides that correspond to a specific amino acid or stop signal during protein synthesis is known as genome, codon, genetic code or bubble. Anyone?
I think codon.
Codon, yeah, correct. So codon is the sequence of genetic code is basically it's a very vague, random, not random, it's generalized word, and codon is, but the sequence of 3 nucleotides is called codon, and it corresponds to a specific amino acid. So each amino acid is coded by a sequence of 3 amino. Each amino acid is coded by a sequence of 3 nucleotides, and these 3 nucleotides in the particular sequence is called codon. So what is a codon? So a codon is a sequence of 3 DNA or RNA nucleotides that corresponds with a specific amino acid or stop signal during protein synthesis. So these are the codons. Let's say GCU, guanine, GCU. This is RNA. So that's why we see uracil. We know that in RNA uracil is present, and in DNA thymine is present. So GCU is one codon. It's a sequence of 3 nucleotides. It's one codon, and this GCU corresponds to alanine, this amino acid. So for ACG, this is another adenine, cytosine, guanine. This corresponds to threonine. So each amino acid has codons. There can be even 2-3 codons that can correspond to one amino acid. And this UAG, UGA and UAA, this corresponds to the stop codon. So the codon is the correct answer. It's the sequence of 3 DNA and RNA nucleotides, and the DNA. So basically there are 4 nucleotides, right? GCT, AT. So 4 nucleotides, and the language of proteins there are 20 amino acids. So each codon corresponds to a single amino acid. Like each codon corresponds to a single amino acid, but each amino acid can correspond to a variety of codons. Like, if you understand what I'm trying to say is, ABC, example, ABC corresponds to one amino acid A, amino acid 1, but amino acid 1 is also made by DEG, if that makes sense. So each codon corresponds to one amino acid, but one amino acid can be made by different codons. So that is why the genetic code is described as degenerate. If you see the last point here, it's degenerate or redundant because a single amino acid may be coded for by more than one codon. More than one codon can code for a single amino acid. Okay, and so when codons are read from the nucleotide sequence, they are read in succession and do not overlap with one another. Okay, so this is the strand, this is the DNA, this is the DNA strand, this is DNA, that's why there is thymine. This is the strand to be transcribed, then transcription happens, RNA is formed. See, T is converted to U because RNA does not have thymine. And from here, if you count, AUG is the start codon, which is methionine. The amino acid methionine corresponds to the start codon. Every time you need to start protein synthesis, the start codon has to be there. This is methionine. Then comes AAG, which is lysine. Then comes UUU, which is PHA, and then UAG, which is the stop codon. UAG, UG and UAA, I guess. These are the stop codons. This is what the 3 nucleotide sequence is called, codon. The next question is, which one of the following statements is not true about protein structure? Anyone? Anyone willing to answer this?
No.
Okay, so okay, no problem. So which one of the following statements is not true about protein structure? The secondary structure of proteins alone gives the protein its specificity to the target compounds. Now, a protein, as I was telling in some other question, proteins have 4 different levels of structures: primary, secondary, tertiary, and quaternary. And this statement, the primary structure of proteins is merely a chain of amino acids connected by peptide bonds. It's a true statement. The third statement, tertiary structure of protein is its 3D conformation and is sensitive to chemistry and temperature. True statement. quaternary structures are the biological units of multiple protein domains. True statement. The second statement is not true, and I'll tell you why. So these are the 4 levels of structure of protein. This is the primary structure. These are the different amino acid residues, and this is the secondary structures. There can be alpha helix, there can be beta sheet. This is the alpha helix structure. So this is the secondary structure. How these are each amino acids, how these are made into helix structures. Then comes the tertiary structure. These strings, they fold upon themselves to form the tertiary structure. So that's why it's a 3D conformation. And then the quaternary structure comes when different subunits of polypeptides, different polypeptides. See, these are yellow. There's this yellow polypeptide. There's this red polypeptide. When different subunits come together and a protein is formed, that's the quaternary structure. So quaternary structure is the 3-dimensional arrangement of the subunits in a multi-subunit protein. So there are some proteins which are single subunit. There are proteins which are multiple subunits. So the specificity of the substrate of the protein structure is determined by the tertiary structure. So the tertiary structure is the one that determines the specificity of the protein to the target compound. Secondary structure does not determine. So each protein has a very specific shape due to their tertiary structure formation, and this plays an important role in the lock and key mechanism of the enzyme. So this string has taken up this shape, but this string could have also taken up some other shape. This string could have also taken up some other shape. So depending on the shape, the target compound, the target molecule will come and attach to it or not. The shape will basically who's joining? So the target compound will attach to it or not, that depends on the shape. So if you guys know about the lock and key model, it's very basic. The lock and key model is basically said in respect to enzymes. There's an enzyme, there's a substrate. The enzyme needs to have a particular shape and the substrate will have exactly a complementary shape. If that happens, then only the substrate will fit into the enzyme like a key fits into the lock. We use lock and key every day in our daily lives. We lock doors and stuff. So have you seen how that only one particular key fits into the lock and opens it? If you try any other key, it will not fit properly into the lock. So that's exactly what is said here. When enzymes are what? Proteins only, mostly proteins only. So there's an enzyme, it has a particular shape, and there's a protein, the substrate, which has exactly a complementary shape. For example, puzzles. If you have solved puzzles like puzzle blocks, if you have solved, you'll see how one puzzle square fits into the other square when they have complementary shapes. That's exactly what a lock and key model says. So when the tertiary structure basically forms the 3D conformation in a protein. So the tertiary structure, if it has a very particular conformation, that is only the time when the substrate or with the target compound will come and sit on it. So everything that with target compound will sit on it or not, that depends on the tertiary structure and not the secondary structure. That was why the second statement was not true. The second statement was false. So the primary structure deals with the sequential arrangement of the amino acids. So this does not determine if the target will come and sit to it or not. The quaternary structure deals with the association of several protein polypeptide chains. Several subunits come and form a protein. So this is closely related. This basically determines the function of the proteins, but it still does not determine. It might have a role, but it's still not the only thing that determines the target compound specificity. And the secondary structure is basically helps us to predict the tertiary structure on the arrangement of the amino acids, but still only based on the secondary structure we cannot say if we only have a string like this, will we be able to say if the target protein will come and target substrate will come and bind to it or not? No. So it's the tertiary structure that is the most important in determining whether the target compound will come and bind to it or not. So that's why the tertiary structure is in its 3D conformation, sensitive to chemistry and temperature. This structure is the one that determines if the target structure will come and bind to it, target compound will come and bind to it or not. Next is dash and dash use inorganic and organic chemicals as source of energy respectively. Anyone?
Option B, ma'am.
Chemolithotropy and chemoorganotropy, right?
Yeah.
Yes, you're correct. The option is chemolithotropy and chemoorganotropy, and this is why chemolithotropy is basically the oxidation of inorganic chemicals for the generation of energy. So this process can use oxidative phosphorylation like aerobic and anaerobic respiration, but the substance being oxidized is an inorganic compound. Just remember that chemolithotropy is inorganic. That's what was mentioned in the first. Inorganic is chemolithotropy, and chemoorganic organotropy is organic. It's in the name only. Organo-organic. So chemoorganotropy is a term used to denote the oxidation of organic chemicals to yield energy, and this process can be performed in the absence or even presence of oxygen, and there can be various byproducts. So this is the chemoorganotropes and chemolithotropes difference. So just remember, chemolithotropes inorganic, chemoorganotropes organic. That's source of electron donor, inorganic, organic. So for oxidizing compounds, since the lithotropes they go for inorganic compounds, these are hydrogen sulfide, ferrous iron nitride, and for organotropes these go for organic compounds. So these are sugars, fats, and proteins. And for examples, we have microbes, bacteria, and archaea. There are bacteria and archaea that can utilize inorganic substrates like this. And for examples, and obviously bacteria and archaea can easily utilize sugars and organic compounds. So she was right. The correct option is chemolithotropy and chemoorganotropy. The next question. Also, so this is another schematic that I showed here to show how the differentiation is done. So soil microorganisms, there is an energy source. If the energy source is light, photo will come in. And if the energy source is light, those are called phototrophs. If the energy source is chemical, that's called chemotrophs. This is very basic. Then from that, if CO2 is being utilized, then it is called photolithotrophs. It's an inorganic thing. And if organic compound is being utilized, it's called photoorganotrophs. Similarly, in case of when chemical things are being utilized, if CO2 is being utilized, it's called chemolithotrophs, and if organic compounds are being utilized, these are called chemoorganotrophs. So that was the answer. So next we move on to the next question. Most energy rich compounds contain high energy bonds having the following property. Primarily phosphorylated compounds, delta G gives energy less than 30 kilojoule, phosphate attached by ether bond, none of these. So I'm sorry, I showed the answer only. So these will be the primary phosphorylated compounds, and one of the prime example of such a high energy compound is ATP, adenosine triphosphate. So see, so these high energy compounds are also called energy rich compounds, and these compounds are present in the biological system, and that when hydrolyzed can produce free energy that is greater or equal to that of ATP. So ATP is a very important compound when we are talking about biological cells. So ATP is everywhere. ATP is the most important prominent compound that gives energy, and low energy compounds have an energy yield of less than this. So high energy compounds, high energy bonds are basically found in most high energy compounds, and these like how ATP is hydrolyzed to ADP, adenosine diphosphate. This has 3 phosphate groups, so triphosphate. This when hydrolyzed gives adenosine diphosphate, which has 2 phosphate groups, and inorganic phosphate. 1 phosphate gets out and diphosphate is formed. So these ATP groups, these are very high energy. These are very high energy bonds, and most of the high energy compounds, even except ATP, it has been seen that most of these high energy bonds have phosphate groups, and that's why they are also termed as high energy phosphates. Okay. So this is the last question that I'll be doing this day. Are the following statements true? DNA encodes the genetic information, which is a template for mRNA, which in turn is template for assembly of amino acids, and this is known as central dogma. Anyone?
True, ma'am. Both of these.
Both are true?
Yeah, DNA encodes the mRNA. Correct. Yes, ma'am.
Okay. So yes, so both of these are true. So the central true. So the answer will be B. So this is the central dogma. DNA, which is a double-stranded molecule, it is replicated, and DNA again, like DNA comes from, like it follows a semi-conservative mode of replication, and the strands are separated and replication occurs. Then transcription gives rise to RNA, mRNA, messenger RNA, and this messenger RNA then works with the ribosome to form the protein that is called translation. And in some cases RNA can also reverse transcribe to DNA. If you have heard of retroviruses have this facility. They have the RNA that can reverse transcribe to the DNA, which is also this is also a very important technique that is used in labs. If you have heard of RT-PCR, RT-PCR is reverse transcriptase polymerase chain reaction. Polymerase chain reaction, PCR, is basically nothing. You are amplifying the DNA. You are replicating the DNA and amplifying the DNA. RT-PCR is when you are taking the main the beginning sample is RNA, and you are making DNA from that. So sometimes what happens, people isolate RNA samples from cells or organisms, whatever they want to. They isolate RNA samples, then they conduct RT-PCR, they or reverse transcriptase PCR, which is they make DNA from that and then they amplify that DNA. So this is also a very important molecular biology technique. Maybe you can read about it if you're interested. It's called reverse transcriptase PCR. So these are the questions that I wanted to cover today. It was a very long session. So okay, Shiny, can you please repeat this? Shiny, if you are here, can you can you let me know which question this was? Actually, I couldn't see the Google Meet chat. If you're still here, if you can tell me which question you wanted me to repeat, I will repeat it. Are you here? Okay, she's not here, I think. She's here. Shiny. Just let me know if you want me to repeat any questions. Was there any question that you want me to repeat or you didn't understand, or you want to get more information? There are questions which are very theoretical, like very information based, like bacterial cells and archaeal cells walls. These things you'll read and you'll forget. That's very normal. So you have to go through Google and search about it. But there are questions that are very conceptual, like the central dogma, the blue white screening, the PCR. These things are very conceptual. If you read about it one time and if you understand it properly, I promise you, you will never forget, and you might be fascinated by it also. So those things I would ask you guys to put more focus on those things, because you read it once and it's a concept and you understand it and you never forget that. Knowledge based theoretical questions are not my favorite also. So those things you have to, like the Anton van Leeuwenhoek question, like we have all read that in school also, but we still don't, sometimes we don't remember who was the first person to have seen bacteria, microorganisms under the microscope. Even I thought it was Robert Hooke then, but then I checked it was Antony van Leeuwenhoek. So these are very memory based questions. Otherwise, thank you, Sneha. That's very nice of you. You're too kind. Shiny, if you're here, you can let me know the question, or maybe we can stop the session, because it was a long session and I'm pretty sure people are tired. Anybody here who wants me to repeat any concept from what we discussed today? Okay. You can also write in the chat box, or you can unmute yourself and say. Okay. Shiny, if you still want me to repeat, maybe you can mail me which question you want me to repeat, or you can just write in the chat box. I'll repeat it once, or anything that you would like. Okay. Anyway, thank you so much, guys, for staying with me for 2 hours. I know it's a difficult thing to stay 2 hours at a stretch in an online. Yes, anybody?
No, ma'am. Just unmuted myself to say thank you.
Thank you so much.
Also.
Thank you so much. Thank you so much. Thank you guys for interacting. I know online sessions are really difficult because we don't get to see each other. Interactions are very important. So thank you for taking the time out, taking 2 hours out of your Friday night to come and listen to this. I hope I didn't bore anyone. And if you have any questions at all, please feel free to mail me. You can find. Thank you so much. Thank you so much, Venisha. We'll meet again next Friday, and do not forget to submit your assignments for this. I think 8th February is the deadline, so do not forget to submit your assignments, and once the due date is over, maybe we can discuss those assignments also next Friday. You need I cannot discuss those assignments right now because you are still yet to do that. Once you finish those assignments and submit those, I can discuss those in the coming week. But don't forget, 8th February is the deadline. I think I saw 8th February. Just check in the NPTEL page and do it on time, okay? And we'll discuss that afterwards, and maybe we'll have an interactive session. Maybe we'll do it again. Thank you so much. You can leave the meeting if you don't have any question. Thank you, Sneha. Thank you, Venisha. Thank you. You can leave the meeting. I'll close the session for today. We'll meet next Friday, same time, 6 p.m. and same link. Okay. Thank you. Bye.
Thank you, ma'am.
Thank you, Prachi. Please don't call me ma'am. Aishi is fine. Thank you. Thank you. I'll close the session. Shiny, you're still here. If you want me to repeat any question, I can see you have written in the chat box, but I could not see the chat box because I was sharing my screen. So if you still want me to repeat, I'm here, or else we can discuss next Friday. Next Friday. Okay. I close the session. Thank you. Bye bye. Have a nice Friday and have a great weekend. Bye.